Good morning, my name is Mike Watt. I'm a scientist at Scion and I'd like to talk to you today about the use of hyperspectral imagery within forestry. This is a very collaborative project that has involved a lot of scientists. The scientists highlighted in bold are international researchers who include Pablo Zarco Tejada, who's one of the leading hyperspectral researchers in the world. Hyperspectral imagery is predominantly used to characterize reflectance across the electromagnetic spectrum. The displayed figure shows reflectance of vegetation across the spectrum. You're no doubt familiar with the visible spectrum that includes blue, green, and red, and the peak that you can see in the bottom left-hand corner shows the higher reflectance of vegetation within the green band. This increased reflectance within this band is why vegetation appears to be green to our eyes. Hyperspectral imagery has many more bands with wavelengths above the visible spectrum. Compared to the visible bands that cover 400 to 700 nanometers, the remainder of the spectrum goes from 700 nanometers to 2,500 nanometers. These invisible bands include the near-infrared band that can be used to characterize cell structure, and the shortwave infrared bands that can be used to distinguish water content in leaf biochemicals. Hyperspectral imagery has been used for spatially describing a wide range of properties. These variables include grass cover, leaf area index, and a wide range of minerals. Within plantation forestry, the main applications that hyperspectral imagery is used for include species identification, nutritional deficiencies, water stress, and disease detection. The pixel size of these spatial representations depends on the platform that is used to collect the imagery, and typically ranges from 1 meter for UAV data to 30 meters for data collected from satellite. We have used hyperspectral imagery to detect nutrient deficiencies. Application of nitrogen and phosphorus have been found to produce significant growth responses in forest stands. However, the detection of these nutrient deficiencies using traditional methods generally involves some form of foliage collection and analysis, which is costly and time-consuming. We set up a trial to determine if hyperspectral imagery could be used to detect nutrient deficiencies. We created a range of deficiencies within 1-year-old radiata pine through providing plants with a factorial range of N and P concentrations. After a number of months, the plants started to respond to these treatments, and you can see the plants with high N and P on the left of the image, which are very dark green, and the very deficient plants, which receive no N or P, on the far right, which have very light coloured foliage. The photo on the left shows the setup we used to acquire the hyperspectral imagery. We placed the hyperspectral camera in the middle of the crossbar that sits above the conveyor belt and ran the trees underneath this camera, which produced the imagery given below. The hyperspectral camera that was used was a Spectrum FX10, which can be used to capture reflectance from 448 bands across a spectral range of 400 to 1,000 nanometers. Compared to this data, you typically get 3 to 4 bands from a multispectral sensor. This slide shows variation in the hyperspectral canopy reflectance between the 5 treatments. As you can see, there are significant differences in reflectance that are particularly evident in the magnitude of the green peak, which occurs around 550 nanometers. A greater proportion of green light is reflected by the control trees with poor nutrition at this wavelength. There are also differences in higher wavelengths between 550 to 650 nanometers. Within the invisible wavelengths, there's also a red shift in the red edge, which is the wavelength range from 680 to 750 nanometers. You can clearly see that this leftward shift in the red edge for the control treatments relative to plants subject to high nutrition treatments such as N1P1. Hyperspectral imagery can be used to derive many indices from the data. Indices typically combine wavelengths with reflectance that vary significantly between the treatment with wavelengths that are insensitive to the treatment. The Carter index is shown here to illustrate the concept. The reflectance at 695 nanometers on the numerator can be seen to differ markedly between the control and N1P1 treatments, as illustrated by the 2 upper horizontal lines. In contrast, the reflectance for both treatments at a wavelength of 420 nanometers is virtually identical. Inclusion of an invariant wavelength can be used to control for other factors that affect reflectance. Using the hyperspectral imagery collected in this experiment, we've developed strong relationships between foliage nitrogen and many indices, including the Carter index, as illustrated by the two figures shown here. We have also made robust predictions of foliage phosphorus when data was constrained to the p-limiting range. Strong relationships have also been developed between indices and photosynthesis. Changing gears a little, I would like to discuss the use of hyperspectral imagery to detect wilding pines. Wilding pines are a major economic and ecological issue in New Zealand and occupy 2 million hectares, which is larger than the plantation estate. Scion has developed an effective herbicide treatment for controlling wilding pines, and this is proactively being used by DOC. Monitoring of control is necessary to determine whether repeat herbicide applications are required, and hyperspectral imagery could provide a useful means of monitoring the efficacy of wilding control. We ran an experiment 2 years ago in which 39 Pinus contortus seedlings were treated with 2 herbicides that included the systemic herbicide triclopyr and the contact herbicide dicot. 13 of these trees were set aside as control plants. The plants were about 1.5 metre high at treatment, and one of the treated plants is pictured. Measurements were made prior to treatment and for 5 weeks following treatment. This slide shows the visual changes in herbicide damage to the trees through time, starting at week 0 at the top and going through to week 5 at the bottom. As expected, there were no changes in foliage colour for the control plants. Visual impacts from triclopyr were first evidenced 7 days after treatment and intensified over time. The visual impacts from the contact herbicide dicot were evident 1 day after treatment and strongly intensified over time. The hyperspectral index, physiological reflectance index, which is a useful predictor of photosynthesis, was extracted from the hyperspectral imagery. Changes in this index over time for the 3 treatments is shown in the figure on the right. The key finding from this analysis was that physiological reflectance index was significantly different between the control and the triclopyr treatment 1 day after treatment. This preceded the detection of visual symptoms which were not evident for the triclopyr treatment until 7 days after treatment. One of the most relevant potential applications of this finding in New Zealand forestry would be the pre-visual detection of red needle cast. If this disease could be detected by hyperspectral imagery from, say, a satellite before it appeared and quickly treated, then the impacts on growth may be greatly mitigated. This is further illustrated by a study on olive trees where the aim was to use hyperspectral imagery to detect the disease Xella fastidosa. The top slide shows the symptomatic trees in dark blue and the asymptomatic trees as unfilled shapes with both categories determined from visual interpretation. The trees that are not coloured in red are those that are not showing any symptoms but were identified as having the disease through molecular analysis. In the bottom image, a model that was developed from hyperspectral and thermal imagery was used to predict whether trees were infected. The key point to take from this image is that all of the trees that were only detected by molecular analysis were also detected by hyperspectral imagery. In summary, using small radiated pine, we've developed strong relationships between hyperspectral indices and photosynthesis and foliage, nitrogen and phosphorus. We plan to scale these predictions up to 4-year-old radiata pine within the accelerated trials over this financial year. We have pre-visually detected the impacts of herbicides on small wildings using hyperspectral imagery. A 3-year-old trial has been established to detect red needle cast from hyperspectral imagery. This could provide a useful stepping stone towards moving towards pre-visual detection of red needle cast in nurseries and the field. Thank you for your attention. I look forward to your questions later.